Well, everywhere we look, we see this incredible complexity, this order, this harmony, that if you recall, Kepler was saying that is what science is all about. Science is involved in looking at this order and the harmony that God has created for us. But when we reject the designer, then you're left with this deep problem associated with the disconnect between a random chaotic box and all the order that we see, and the beauty and the grandeur that's associated with it. Well, we're going to look at a few of these. What do you suppose this is? Joshua, you know what this is. What is it?
It is a blood clot.
It is a blood clot. What are the red things there?
Red blood cells?
Red blood cells, that's exactly right. What are the white things, the white strands?
Oh, is this
Fibrin? Called fibrin.
Yes, that's exactly right. There's strands of fibrin. I'm impressed. Blood clotting is an incredible thing. Have you ever thought about it? What an amazing thing God has given to us in this little system that we just ignore called the clotting of the blood. Or do you think about this? I mean, what would happen? We get cut, and we have to ask ourselves the question, why don't we bleed to death? Actually, I've got a friend who can explain this a whole lot better than I do, and so I'm going to have her explain this for you.
I cut my finger this morning, and it's bleeding. But if I put this Band-Aid on, it'll stop in a while. Did you ever wonder how it happens? I mean, does blood just stop because that's what it's supposed to do? Why doesn't our blood clot before we get it cut? I guess we just die then, because all our blood would harden up and stop flowing. Did you ever wonder why? Blood clotting is a very complex process involving thousands and millions of triggers that have to act just perfectly with one another to create the final outcome. Let me see if I can tell you how this works. First, you get a little cut like mine. Imagine you're in my bloodstream. There's a bunch of traffic going on, and pretend you're floating around with a cajillion other red blood cells, all with oxygen backpacks. Everything slows down when you get near the cut. This is called vascular constriction. In short, your body limits the flow near the cut because it knows something is wrong. And of course, you feel pain. So, a protein in your body called fibrinogen arrives on scene. Fibrinogen is primarily responsible for stimulating platelet clumping. Thrombin essentially cuts off the ends of the fibrinogen. Platelets clump by binding to collagen. Upon activation, platelets release adenosine 5-diphosphate, ADP, and TXA2, which activate additional platelets, serotonin, phospholipids, lipoproteins, and other important proteins for the coagulation cascade. Activated platelets change their shape to accommodate the formation of the plug. Oh, sorry, I digress. Anywho, this complex thing called the Stewart factor converts prothrombin to thrombin, thereby converting fibrinogen to fibrin. By the way, the Stewart factor wasn't active until it was activated by the Christmas factor. Okay, there's a lot more to this process, like this goes there, and binding, recepting, who knows what. It's very complicated. But the net result is a clot. Stops the bleeding, cut heals, clot dissolves, you're on your way. Isn't that neat?
Isn't this an amazing system? Now, how long do you suppose that we took in our evolutionary process before we finally got the clotting system right? How long do you suppose we could last without a clotting system? Not very long. And especially the many generations it would take for us somehow to figure out all those factors. Do you know what happens when one of those doesn't work? Hemophilia is a result of one of those factors not working. What an incredible thing that God has given to us. in blood clotting, which we probably just ignore because we expect it to happen. Okay, let's examine this reality then. Let's begin to look at Carl Sagan's statement, and we'll take his statement as the outline from which we're going to examine this theory. Sagan said that evolution is a fact amply demonstrated by the fossil record and by contemporary molecular biology. Well, if that's the case, if the fossil record and contemporary molecular biology are two things that demonstrate and support the fact of evolution, then let's simply take a look at those and see what reality says. We'll begin with molecular biology, and then we'll talk about the fossil record. What we're going to do is, first of all, begin with Charles Darwin's own statements about his theory. What it is that he laid out as the criteria by which one could reject his theory. Let's look at this first one. Darwin stated, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Do you understand the criteria he's laying out there? Because if gradual Darwinianism, his theory that said over a long period of time, organisms have these very slight changes, these very slight modifications, and that is what has led to the deep complexity that we see today. So what he is saying, he said, if you can demonstrate that there is a complex organ that could not have come into being by that slow, gradual process, then he said, my theory would absolutely break down. So let's take a look at that. We're going to ask Dr. Meyer again to examine part of the reality that exists when we take a look at Darwin's time and his theory, what he was saying in terms of what we're seeing today.
In the 19th century, biologists thought that cells were simple, homogeneous globules of plasm. Now, with that kind of understanding of life, it was easy to imagine how life might have arisen through a few simple chemical reactions, and the origin of life was no big problem. Post-1953, with the discovery of the structure of DNA and also the information-bearing properties of DNA, we can no longer look at the cell the same way. And what we're now discovering is that the cell is chock full of exquisite miniature machines, little turbines, little rotary engines, little walking robots that cart materials from one end of the cell to another. Now, in our experience, all of those kinds of entities are produced by one cause and only one cause, and that is intelligence. And that, again, challenges materialism, because the materialistic worldview of the 19th century said there's two fundamental realities, matter and energy. What we're now discovering is that there's a third fundamental reality, and that is information. And everything we know about information is that it must come from an intelligent cause. So the presence of information in living systems suggests an intelligence that pre-exists us, that is responsible for all that exquisite nanotechnology and information.
Okay, so what Dr. Meyer is talking about here is this idea that what has happened in terms of the technology that we have now is that we have begun to take a look at that inner cell. Remember Paley's argument. If, he said, you were to find a machine, a complex machine, such as a pocket watch, you would understand instantly, you would know, it's plain to you, that what you have in your hand is not something that has just gradually come about as a result of some natural force. It's very clear to you. This has some sort of a design, and it carries a purpose. Now, as Dr. Meyer was saying, in Darwin's time, the cell appeared to be a hidden little black box. It wasn't a machine, it was just some little living glob, and somehow it had the potential within it to be able to mutate and to change. But what we have today is the technology to actually look inside that cell, and what we find is incredible. We're going to return to unlocking the mysteries of life and look at what that inner cell looks like and the machinery and the incredible magnificence of what is going on inside that cell. Now, what I want you also to notice is that at the end of this, there will be a man by the name of Dr. Dean Kenyon, and he will be making a statement as the result of seeing this complexity. And in order for you to understand the importance of this man's statement, you need to understand that for years and years, Dr. Kenyon was one of the leading scientists and proponents of his theory in trying to describe how the molecular structures and everything comes together. He was a proponent of a theory called chemical evolution. And you might be interested to see his statement about the grandeur of what is now to be seen within the living cell.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
What an amazing thing going on inside every living cell. the gentleman at the end, Dr. Kenyon, beholding the reality of what was going on within the cell, recognizing that his own theory, faced with the reality of the deep complexity, he used to believe for years and years that there had to be some random process there because there couldn't be a creator. And so his theory that some chemical attraction would bring all those things together. The more he began to look at the cell and the complexity, he realized that was impossible. And now that we understand what is really going on inside there, Dr. Kenyon recognizes and understands that this is the result of intelligent designer. That's the way we should respond when we gaze into the box, when we gaze and see the plain evidence that God has given to us. You know, the problem is that doesn't always happen. Man doesn't always accede to the reality of what is plain to them. Dr. Francis Crick, a brilliant man, the co-discoverer of DNA, when he began to gaze into the cell and began to understand the deep complexities of what we have there. In fact, if you really want to understand how complex this issue is, it has been stated that just for the single amoeba, the information that is found within the amoeba to provide all of the DNA structure would fill 1,000 sets of Encyclopedia Britannicas. Amazing complexity. So when Dr. Francis Crick is peering into the microscope, and it is beginning to dawn on him the deep complexity of what we see here at the fundamental level of life, he becomes convinced that this could not have happened by chance. He says, an honest man, armed with all the knowledge available to us now, could only state that in some sense the origin of life appears at the moment to be almost a miracle. So many are the conditions which would have had to have been satisfied to get it going. So what does Dr. Crick do? Does he